Hi, I'm Andreas Puja at Bell Communications Research, or Bell Core.
Hello, I'm Werner Stützler from the University of Washington. This film is about visualization of quantitative information. There are two major classes of tools for analyzing and comprehending quantitative data: numerical summaries and graphical presentations. The former, numerical summaries, have certain disadvantages. They require assumptions and are therefore not always meaningful. For instance, means and standard deviations summarize bell-shaped distributions of data well. But what if the distribution is skewed or multimodal? Visual presentations of numerical information, by contrast, do not require assumptions. Whether bell-shaped or multimodal, it is apparent from a plot of the data. A problem in visualizing quantitative information is that data tend to be high-dimensional. Many aspects or dimensions are often considered simultaneously. A common approach to visualizing high-dimensional data is to increase the complexity of pictures. That is, one tries to pack all information into a single picture. The result is pictures which are suitable as information storage devices or pictorial encyclopedias rather than tools for exploration and comprehension of data.
There are two keys to the visualization of complex data: interactive focusing and linking multiple views. Combination of these two principles offers a solution to the problem of visual overload. Instead of trying to cram all the information into a single view, we need to provide tools for quickly generating multiple views, each homing in on a different aspect of the data. Multiple views, however, should not be regarded in isolation. They need to be linked so that partial information can be integrated into a coherent image of the data as a whole. These principles, focusing and linking, are the two major themes of this film. We'll have many examples of interactive focusing later on. For the time being, we concentrate on multiple views. There are two families of techniques for showing multiple views: sequential display and simultaneous display. An example for sequential display of multiple views is 3D rotation. The connection between displays is established by continuity of motion. In the example shown here, we recognize quickly that the three-dimensional data points basically lie on a curved surface, with the exception of an interesting kink, colored red, and an exceptional point, colored yellow. These data arose from an experiment with a laser device at Bell Core. With 3D rotations of the data, engineers were able to discover peculiarities in the performance of the laser. Later on, we'll show generalizations of 3D motion to higher dimensions. Alternatively, we can display multiple views in parallel and connect them by painting. Here, we see a map of the US showing some 300+ metropolitan areas in the upper plot. Below, we show a scatter plot of two indices which measure aspects of livability in metropolitan areas. In a blow-up, we indicate the meaning of the axes. Horizontally, we plot a climate index where left is harsh and right is mild climate. Vertically, we plot a housing cost index with inexpensive places low and expensive places high in the plot. We notice a cluster of metropolitan areas with mild climate and expensive housing in the upper right part of the scatter plot. We might wonder where this cluster falls on the map. A natural hypothesis would be that it corresponds to California. To confirm this hypothesis, we paint the cluster in the scatter plot. The points on the map corresponding to the same metropolitan areas change color. They are indeed in California. This establishes a connection between the two views. Our hypothesis was correct. Moreover, we learned that these metropolitan areas do not lie just anywhere in California, but exclusively on the Pacific coast. Conceptually, we may summarize what we just saw as follows. The map and the scatter plot convey two data dimensions each. By linking the two plots, we perceive four-dimensional structure. For example, we showed via painting where expensive housing and mild climate can be found in terms of geographical location. We emphasize that linking views is crucial, since seeing separate 2D views is not sufficient for reconstructing 4D structure. From a user's point of view, we like to think of painting as an operation on the underlying objects, rather than their visual presentations on the screen. That is, we urge you to think of the operations we just performed as painting coastal California, rather than painting some dots in a scatter plot. With this model in mind, it is natural to expect that painting in one view will cause other views to reflect changes. A color change is not particular to a window, but to the underlying objects. We should now be ready for extensions of the same ideas to other types of views.
Here is an example of how this framework applies to the visualization of discrete data. The left plot shows the latitudes and longitudes of 300 odd English villages. The plot in the upper middle shows the villages classified according to the pronunciation of the word sheath. Similarly, the plot in the upper right shows the villages classified according to the pronunciations of the word sweat. Each cell in a table represents those villages using a particular pronunciation. In the case of the two-dimensional table in the lower half, a cell represents those villages with a particular pair of pronunciations for the two words. The rectangles inside the cells encode the number of villages which use a certain pronunciation or pair of pronunciations. If we now paint the largest cell in the two-dimensional table with green color, we can see where those villages are located in the map. The map shows that villages with this pair of pronunciations occupy a fairly contiguous area. We have thus found a regional dialect. Note that the largest cells in the one-dimensional table above end up partly colored because the painted cell in the two-dimensional table below is the intersection of the corresponding cells in the one-dimensional tables. Next, we paint the second most populated cell in the two-dimensional table with blue color. Of course, we can also paint villages in the left one-dimensional table in the map and in the right one-dimensional table. In the end result, we painted the union of several cells and some other villages on the map.
This illustrates another use of painting, namely as a way of formulating database queries. We just gave examples of forming unions and intersections. It is possible to express any set theoretical clause as a sequence of painting operations in one- and two-dimensional plots. A major advantage of formulating queries via painting is that they can be formed based on visual information about what's there. The pronunciation data are an example of spatial data, as the pronunciations are a function of the villages and hence geographic location. Another frequent type of data are time series, where the observations are a function of time. Here's an example. The observations consist of almost 900 measurements of tidal levels, taken at 45-minute intervals, covering about 27 days. In the picture shown here, the tidal levels are plotted against time. We would expect to see some near periodicities, which have to do with high tide and low tide. This is currently not visible due to unsuitable scaling. We now rescale the plot by stretching the horizontal axis and shrinking the vertical axis. The perceptual effect is drastic, and we assure you that nothing but a scale change has happened. We now expand both axes somewhat to have a closer look at the near periodicities. Since much of the time series has to be clipped at the left and right boundaries, we inspect the data by panning sideways. At this point, we make an interesting discovery. An outlier of sorts appears invisible on previous scales. We tweak the scale one more time to focus on the outlier optimally. We have here an example of data exploration through scaling and panning. Both scaling and panning are instances of interactive focusing, one of the two main themes of this film. We now return to the theme of multiple views, linked both through motion and painting. First, however, we have to mention a manipulation which is common in time series analysis, namely relating the present to the past. We do this, for example, when we try to predict present from past observations. A simple tool for such purposes are so-called lag plots. For every observation, we plot its current tidal level on the vertical axis and the tidal level for an earlier time on the horizontal axis. Here we are looking at the lag 1 plot, where the present level is plotted against the level 45 minutes earlier. Now the lag 2 plot 90 minutes earlier. Now the lag 3 plot 135 minutes earlier, and so on. Some interesting patterns appear in these lag plots, and we will explore them shortly. Back to the lag 1 plot. We will now try to predict the present tidal level from the ones 45 and 90 minutes earlier. We do this by rotating the past two levels horizontally, holding the vertical axis, that is the present level, fixed. The data seem to lie on a steep disk in three-dimensional lag space. We will stop when we catch a view over the edge of the disk. An almost perfect straight line, with the exception of a few outliers. This tells us that the present can be predicted from the past two observations via a simple linear equation. Let us now go back to a lag plot of the present versus about 3 hours earlier. It features a peculiar double loop pattern which we may want to explore. A natural question is to ask how it relates to time, that is, what do the orbits look like in this patterned point cloud? Here is the answer. We brush time intervals in the time series plot below and observe fragments of orbits in the lag plot above. The current time interval shows orbits which run along the periphery of the point cloud. Next, we pick a time interval in the right half. It has orbits which stay in the interior. The orbits move in double loops and shift over time. This gives qualitative indications of facts which can otherwise only be detected with spectral analysis. The double loop indicates that there are two basic frequencies, one double the other. The shift in orbits is due to the presence of very low frequency components.
Here is a data set which arose from an experiment in speech recognition. A native Dutch speaker spoke 11 Dutch vowels on a tape. From each of the 11 vowels, 6 short time fragments were extracted and digitized, resulting in 66 data vectors. These vectors were put through spectral analysis and ultimately reduced to vectors of dimension as low as 5. The question is whether the reduced low-dimensional data allow us to classify the vowels. Rather than performing a standard discriminant analysis, we use an exploratory approach, in which we visually search for natural clusters, which we then try to relate to the underlying vowels. We will be quite successful indeed. The plan is to start with 2 of the 5 dimensions, see what we find, and then escalate to 3, 4, and 5-dimensional data rotations, successively isolating natural clusters corresponding to vowels. In the upper plot, we are looking at 2 of the 5 dimensions, while the lower plot shows us basically a visual representation of the experimental design. 11 vowels from left to right, represented by columns of 6 dots per column. Back to the upper plot. We may perceive 2 or 3 natural clusters, the one in the top left being the most pronounced one. Painting it with red color shows in the lower plot that we captured exactly 2 vowels, denoted EY and I. To avoid obscuring in the following views, we remove them by applying an eraser brush in the lower window. Next, we rotate the remaining points in 3 of the 5 dimensions. We perceive a wreath-like structure, which we place in a convenient position for painting. coloring the lower half of the wreath, we find in the lower window that we subdivided the vowels into two groups, with the exception of one misclassification. Having explored three-dimensional structure, we now turn to a tool for the display of four and higher dimensional data called the Grand Tour. What we currently see are Grand Tour sequences alternating with painting of newly detected clusters. The Grand Tour is a direct generalization of 3D rotations. To generate 3D rotations, we show projections onto a moving two-plane in 3 space. Similarly, the Grand Tour shows projections onto a moving two-plane in arbitrary dimensional space. Although human 3D perception does not work well for Grand Tour sequences, there is still a lot to be learned from watching a Grand Tour, as we see now that we are Grand Touring 4 of the 5 dimensions of the Dutch vowel data. Additional clusters corresponding to vowels become readily apparent.
As a final display of the Dutch vowel data, we'd like to show you a so-called Andrews curve plot. Besides being a multivariate plotting technique, it also offers an interesting variation on the theme of linked views. Start with a one-dimensional projection of the data, plotted, say, vertically. This by itself is fairly uninformative, a collection of dots on the vertical axis. However, it gets interesting if we let the projection direction smoothly wander in data space, while tracing the dynamic one-dimensional projections from left to right on a plotting surface. Here is what the result looks like for the Dutch vowel data. 66 curves, 1 per vowel fragment. Each vertical section through this set of curves is a one-dimensional projection. Apparently, there are many projections which isolate natural clusters of curves. Again, we can use painting as a means of verifying that the clusters do indeed correspond to vowels. In what follows, we paint 2 subsets of curves and establish vowel correspondence in a separate window. The intriguing point is that Andrews curves can be seen as a large number of one-dimensional projections, linked to each other by a continuous flow of curves. This ties them in with our theme of linked views of data, although linking is established by altogether different means.
In our next example, we will have a look at an MRI image of a slice of the human head. This is a 3-channel image. For each pixel we have, besides its spatial coordinates, the values of proton density, T1 wave and T2 wave. Thus, there are 5 variables altogether and about 21,000 data points. Each of the 3 images on the left of the screen shows a grayscale encoding of one of the channels, dark for low values and bright for high values. With these plots, we hope to identify anatomical structure. Another way of presenting the data is to discard for the moment the spatial information and regard each pixel as a point in 3-dimensional channel space. The 3 plots on the right are pairwise scatter plots showing proton density plotted against T1 wave, proton density against T2 wave, and T1 wave against T2 wave. Displaying 21,000 points in a scatter plot would result in a picture that is hard to interpret due to the frequent overstriking. We have thus selected a random sample of a few hundred pixels for display. Notice how the points in channel space tend to group into clusters. These clusters have different orientations, and some are very sparse while others are dense. We can summarize the structure using a clustering algorithm. It will organize the data into a hierarchy or tree. Here we see a close-up of part of the clustering tree. The root of the tree is on the left, the leaves are on the right. Each node of the tree represents a subset of the pixels in the image, and its daughters define a partitioning of this subset. This is an overall view of the tree. Due to space constraints, it cannot display any detailed information on nodes, such as the number of pixels. To obtain a more detailed view of selected regions of the tree, we can use a magnifying lens, a little rectangle that is moved over the tree under mouse control. The part of the tree that is under the lens is shown magnified in the window at the top right of the screen. At this level of detail, we can interact with individual nodes. By cascading magnification lenses, we can move in close enough to discern detailed information about individual nodes. Zooming in makes the detail clearer. Make another magnification lens to compare two different views side by side. For a given level of scrutiny, the appropriate type and amount of information is presented. This is the principle of logical zooming. An obvious question is where pixels in a given node of the tree fall in the scatter plots and in the image. Painting allows us to establish this connection. In our example, we have painted the pixels in the chosen node green. Obviously, the cluster we picked corresponds to the brain. Next, we will subdivide the subset by painting one of the daughter nodes in the tree red. We see in the image that we have separated white matter from gray matter. Here is a close-up of the image with gray matter drawn in red. It would be interesting to see whether pixels identified as gray matter occur in other parts of the image. To find out, we will again use a magnifying lens. Zooming in, we notice a thin, contiguous set of red pixels outside the brain in the back of the eye. A reasonable hypothesis is that they correspond to the retina.
Our final example provides another illustration for the use of focusing and linking views in image analysis. What we see here is a Landsat image of part of the Amazon basin around Manaus, Brazil. The wide area to the left of the center of the image is the city of Manaus. The dark river is the Rio Negro. The blue river is the heavily sedimented Amazon. This is a 4-channel image. For each pixel, we have the reflectances in 2 infrared wavelengths and 2 visible wavelengths. We can use a magnification lens with logical zooming to explore the spectra of pixels in different regions of the image. The window which we open near the top of the screen is the magnification window. It shows the spectrum of the pixel we are currently pointing at with the mouse. The spectrum consists of only 3 straight line segments because we have reflectance values for only 4 frequencies. The spectra we saw initially were typical forest spectra, characterized by low reflectance in the visible bands and high reflectance in the infrared. We will now select a subset of the pixels for analysis of their distribution in channel space. We pick some pixels from forest, some pixels from dark water, some pixels from swampland, and finally, a set of pixels along a transect across the river containing dark water as well as heavily sedimented water. Here we are looking at a scatter plot of the geographic locations of the selected pixels. The 3 small clumps are forest at the top, dark water on the left, and swamp at the bottom. The long line of pixels is the transect across the river. We will now use the Grand Tour to look at the selected pixels interpreted as points in 4-dimensional channel space. By searching for properties of the projected point cloud that stay invariant as the projection plane moves, we can find out facts about the 4-dimensional structure. For example, pixels that are close together in all projections are close together in 4 space, and pixels that line up along straight lines in all projections lie on a straight line in 4 space. On the right-hand side of the screen, we now see one of these linear features. In addition, the picture shows 2 roughly spherical clumps surrounded by some fluff. We can use painting to find out where those pixels fall in the map. We now see 2 windows on the screen. The previous projection of the 4D channel space on top and the geographic locations at the bottom. Brushing over the forest pixels in the map shows that they correspond to the upper one of the 2 spherical clusters in channel space. Highlighting the swampland pixels reveals that they correspond to the lower cluster. Dark water is found to correspond to one end of the linear structure in channel space. We now brush along the transect on the map. The very top of the transect is forest. Next comes dark water. By moving the brush along the transect on the map, we see that the spectra of pixels along the transect are mixtures of the spectra for dark water and heavily sedimented water, respectively. This makes sense when we recall that the transect was placed where the Amazon meets the Rio Negro, and thus dark water is mixed with heavily sedimented water. Explaining the spectra of pixels as mixtures is of interest in several applications of image analysis.
Finally, let us summarize. There are two keys to the visualization of complex data: focusing and linking. Combination of these two principles offers a solution to the problem of visual overload. Instead of trying to cram all the information into a single view, we need to provide tools for quickly generating multiple views, each homing in on a different aspect of the data. Multiple views, however, should not be regarded in isolation. They need to be linked so that partial information can be integrated into a coherent image of the data as a whole.